Okay. Thank you, everybody, for coming. It's a great turnout today. It's a real pleasure to introduce my mate, I'm going to say, Alan Wilson from Auburn University. I've connected with Alan when I first came to the C Lab, actually, looking for anyone else in this region that cared about HABs. And it took me less than 5 seconds to find Alan's webpage and all of the cool things that he's doing up at Auburn University. So I'm thrilled that I've been able to work with him over the last several years on many different projects. One of the first things we were interested in was working in looking for different cyanobacteria that are in the mobile tensor delta. So Alan works a little upstream and I work in the coastal areas, in the bays. So at this land-sea interface is a really important place to be. But Alan's research is focused on cyanobacterial blooms and their effect on phytoplankton and zooplankton communities and many, many other things. He is a freshly stamped professor at Auburn University, which is an awesome accomplishment, so congratulations for that. He did his PhD at Georgia Tech in 2006 in Mark Hayes' lab, which is a chemical ecology lab, and was in grad school with our own Dr. Smee. So I'm sure that they have some fun stories to talk about later. Alan is also the RU site director for warm water aquatic ecology at Auburn University, which has been what seems like a huge success. He also runs a very successful meta-analysis class that you should all consider for the graduate students that are here, which has resulted in about 30 different papers published by students as a part of that class. So it's really worthwhile skill to get to know, and I think it's really well received and reviewed by students. So give that a look for the spring. I was just going to say current events that we have going on. We've had massive freshwater pulses into the coastal environment around Mississippi Sound and across here into Alabama. And Alan and I were able, along with a few other researchers here, were able to get event response funding from NOAA. So we're working to try and understand those mechanisms and do a lot of work. So Alan, you might see more of him as we develop this research and try and understand what's happening. It was also a real pleasure to have Alan join us in Cuba as part of our other work that we do. He was well loved in Cuba and was helping with different cyanobacterial issues in the lakes there, and I think that work has also now resulted in some joint publications and things like that, so it's fantastic. Alan's a great scientist, but also a great guy, very easy to work with, and it's been wonderful for my students to interact with him and get to know him. And I think it helps to have somebody that's, nice and easy, level-headed on the team when you're working in event response kind of things. So with that, thank you for coming, and I hope you learn a lot from Alan. He's also going to give a graduate workshop after lunch on fellowships and funding, which he was the NSF program director in the DB program for a couple of years, so he has a lot to offer and information to give you some ideas on how to gain extra funding for your research and moving forward even into postdocs and other things. Thank you very much.
Okay. Yeah, do I need this? I guess they need it for the video. I did approve the video. I don't particularly like seeing myself on video, but I'm going to do the best I can. So thank you, Allison, for giving me the opportunity to be here. I'm so happy to be able to spend time with Allison. Hopefully you appreciate what you have here. She's awesome, and she's been a great collaborator. I wanted to make sure I would be happy to have any students who want to take the meta-analysis class I offer every spring. I do offer folks remotely to connect. You wouldn't be able to formally register, but you could take the class just like everybody else. And we've gotten 29 papers from the students. Students in my class are unbelievable. So if you want to take it, let me know. I'd be happy to get you involved. Yes, I'm psyched to be here. I've never been to Dauphin Island. Today is my wife's birthday, so she was psyched to be able to come down with my kids, and they're at the beach right now. And Allison said the water is great, so thank you. And I have a tendency to like watching the slides with you, so if this will work, I'm going to try to stay close to you. So just as a reminder, I've been at Auburn for, I guess, 12 years now. I'm in the School of Fisheries, Aquaculture, and Aquatic Sciences. I like to show this picture. I don't know if we can pull the lights down a little bit, if that would be possible, just because the pictures I'm going to show I think are spectacular if you get to see the greenness. But these are experimental ponds. We do a lot of aquaculture research. And honestly, I have lots of great colleagues. I love my students, but these ponds are my favorite part of my job. The fact that someone who likes to do field experiments, and we have tons and tons of resources available that are typically for green like this. So it's somebody studies freshwater algal blooms. This is like the perfect environment. So just as a reminder, I do a lot of work in aquaculture. We like to do field experiments. This is one design we've used, and actually we're having an experiment that we just ended in a drinking water reservoir with this particular design. So those are just trash cans that we floated on a PVC frame, filled them up, and did some different treatments. This is the drinking water reservoir where we have an experiment right now. This is just a scale to give you a sense. This is my grad student, Michael, who finished up a couple years ago, and Bree got her master's. But just for their scale, he's about 6 foot. So these are pretty big bags. Okay, so here we go. So I'm interested in the history of science, and so at some point I kind of dug around, looked for the oldest paper about science, or algal blooms in particular. And this is the one I came across. It was published in 1878. There were a bunch of dead livestock that were around this pond or this lake in Australia, and they were linking that to water quality. So again, people have been studying algal blooms for a long time. There's still a lot to be explored, but there is a long history of this. I'm going to show you some pictures. I love taking pictures. If you see me out and about, you'll see me taking pictures of stuff. And so I'm just going to show you some of my favorites. This is an aquaculture pond in West Alabama. I go sample these ponds once a month. This is an aerator. They use this to help keep the water oxygenated overnight, because although during the day this pond is super saturated, tons of oxygen, at night it can go anoxic because everybody's respiring. So they turn these on at night to make sure the catfish don't die. At some point I was looking for a picture of a dead cow in a green pond, and someone in South Africa sent me a picture of a dead buffalo, and you're going to see this picture in a second. It's not as graphic as you probably think it's going to be, but if you put, at least several years ago, if you did a dead cow picture, you see a lot of desert shots, but that doesn't really work well with this kind of context. So I need a green pond. But my colleague sent this picture, and I love when I'm flying, I love to look out the window. Honestly, whenever I travel, I'm looking for green systems. And I think this is spectacular. I mean, these yachts, all of us would beg to have any of these boats, but to think that these boats were in this pea soup is remarkable. But to me, this is art. This is a picture I took when I went to Mexico. I love this picture for a lot of reasons. This bloom of Microcystis and oscillatoria was super, super dense. And as we got further into the bloom, it looked like the algae was actually so dense, it was trying to come out of the water. So, I mean, this is incredible. This is this lake was massive. It took us about 2 hours to boat up to the top of it. And that bloom was impressive. So you've all heard of Lake Erie. Lake Erie struggles with algal blooms every summer. This is a picture I took during my postdoc. I got some pictures looking out, but they weren't quite as impressive. So if you look down, you can see these huge colonies, which typically these are microscopic, but you can see them with your eyes. This is a blade of Vallisneria, so you can see that these microscopic algae are really big and really abundant. Okay. So Allison just saw this picture. She's like, "That's pretty creepy." So yeah. So it's so why am I showing you this picture? So this is interesting. So if you look down here, this little story about Fertilize the World, I got contacted by a photographer for National Geographic that was looking for some green ponds, and I like to talk about our green ponds. So the word got to him. He came down to Auburn to take some pictures of me and my students. Actually, not so much me, but my students, my grad student, and my 2 RU students at the time. And this is my old blue truck. And here's Peter taking pictures of my students. I was so excited to think that our green ponds, my RU students, would be in National Geographic. Sadly, that didn't happen. This is the picture that showed up in National Geographic. Everyone who studies algal blooms has probably seen this picture. If you haven't, you've seen it now. This is spectacular. This shot is unbelievable. This is a boat flying through the water. And just the scale of the bloom, the thickness of the bloom. So pay attention to the year. So this is 2011. And that timing is important because many of you probably heard that they had a major issue in Toledo where they had to shut down drinking water for a few days because of toxin levels. And so this is 3 years later. You know, this happens every year. This is a common occurrence, but this is a big deal. And so for several years, Lake Erie, this particular western basin, has been kind of on the radar for algal blooms. And it's kind of job security for people like me and Allison that these events happen. And I think they're going to continue to happen. I don't see the issues that are plaguing the western basin of Lake Erie being resolved anytime soon. But fortunately for Lake Erie, there was a new spotlight out, and it was on Lake Okeechobee down in Florida. And when I was a kid, Lake Okeechobee was really clear, lots of macrophyte. Apparently now there's lots of algal blooms, lots of agriculture going into the lake. And so this lake had been kind of the hot spot. And part of the reason it was the hot spot was that they were having the water diverted from the lake out to the ocean. And so all that algal material that doesn't particularly like salt water dies. And so again, if you own this fancy boat here, you don't want it to be surrounded by this scum, which is disgusting. So anyway, so this has been taking some of the heat off of Lake Erie. But now, hopefully, all of you are aware of what's happening out around here. They've closed the beaches for several months, basically summer in Mississippi. All the beaches were closed for a long time, which is a big deal. And so what was interesting about what's happening, and I'm sure all of you know this, but I love the spillway over in New Orleans. I would take my RU students down there every summer, and it never had water. It was reliably dry. So just as a reminder, the Mississippi River flows down through this way. And but right here, there's a spillway that if the water gets too high, they can release it to Lake Pontchartrain so it doesn't flood New Orleans, and then it finds its way out to the Gulf. And Dolphin Island is over here. So this system works pretty well. And this is what it typically looks like. This is the Mississippi River. There's the spillway. And then if you look down here, there's lots of roads. There's lots of actual recreational activity out there. People bike, people motorbike, people fly helicopters and airplanes. It's a whole thing out there. And we always would go in the summer, and I'll show you some pictures of my RU students. This is us out doing work in the spillway. So this is where, at least a few months ago, you wouldn't want to be, but that's us collecting organisms in the spillway. This is what it looks like. So this is the Mississippi River side, and that's the side that is always dry, at least had been regularly dry. These are the little pins they pull out to release the water. So this is what it looked like for over 100 days. And again, I had been going down there every year since 2011. Never was it open when we were down there in July. This is what it looked like for 100 days. Just as a scale, I think if you don't believe in climate change, not to think that anyone in here does, but this is just a reminder of what happened. These are the openings. See that the number of bays they open. They used to open all the bays, and now they don't open as many. But they kept this one. The first time that this spillway was open twice in 1 year, and again, it was the time that I knew the summer. And this is just another scale to see how many openings in a particular decade. So if you look at this trend, typically it's 1 or 0 per decade. 70s is a little different, but in our current decade, 5 openings already. To me, this is a warning, and I fear that at some point, maybe in my lifetime, New Orleans will get underwater and won't be able to be dry, which makes me sad. But this is a headline that just came out in the Huffington Post just a few days ago. Tourism on the Mississippi coast has been devastated by water coming in through the Mississippi River down to your coast. And again, it's a remarkable story, but the fact that we go down there every summer, I can't believe that much water had been flowing through there. So just as a reminder, some of you may not know about algal blooms. They like the algae that Allison and I study. They like nutrients. They like warmer water typically. Flushing can be good or bad for algae. It depends if that flushing is just pushing water through. It may be bad because some algae grow slow. But if that flushing is bringing nutrients in, it may be good. But the idea is that if you have high nutrients, high temperatures, high light, you can feed algal blooms. There's a salinity component here. Again, do well in marine systems. Don't grow there. So that's why this event that's on your coast is so unusual. So I'm an ecologist, so just as a reminder that I do think about these top-down factors, and I'm going to talk a lot about that shortly. But just as a reminder, this is just the Trophic Cascade, and the idea is if you have these big fish out there, they eat the little fish, and then that allows these large zooplankton to be abundant, and then they eat the phytoplankton. Ideally, if you have a body to manage and at least in freshwater systems, keep these big fish happy, you would have clear water. That's the theory. And I'm going to show you some examples of that in a second. All right, there's my dead buffalo. Again, I spent so long getting that picture. This is a picture I took. So again, this is the scary picture, why we care about algal blooms. They can kill your pet buffalo. They can kill your fish that you're growing. They can make your water taste like mud, and that's not great. We're dealing with this in Auburn right now. And then some municipalities actually will restrict access. So, for example, if you own a business on a water body, Mississippi, and they say stay out of the water, people stop coming to your store, and that could devastate your family, your community. And then again, this is, I don't know if you all have seen this, but my phone has been kind of blowing up the last maybe 3 weeks or so once this story came out. Dead dogs due to algal blooms happen every year. This happens. Dead livestock, I get calls every year. This year, I think the fact that this got such big press has gotten more people to be worried. I had people sending me samples of perfectly clear water that they were concerned. And so I look at it, and again, there could be stuff in the water that killed their dog, but it's not algae. So I'm like, at least what I study, it's not a problem. But there may be other stuff. But the idea that this has really brought algal blooms to people's awareness, which it's unfortunate for the dogs, but I don't think this summer is unusual, at least as far as I'm concerned. Okay, just a couple reminders of how we kill algae. There's a bunch of different approaches. So there's physical approaches, there's chemical approaches. These are commonly used because they're quick. And then there's biological. So I'm an ecologist, and so what I'm going to talk about today are trying to think about how we could use food web manipulations to control algal blooms. And I think it's possible in some systems. I know it's possible. Okay, so here we go. This is the real part of my talk. Sorry. I just was so excited about the spillway. I just love going down there, and I thought given the proximity of the spillway to y'all, I had to talk about it. So here's the title. So I'm going to talk about both combinations of, let me go back to the title, of how eutrophication, but also consumer adaptations may matter. And I hope that I convince you at the end of this that both prey and consumers can adapt, and they both may matter, and they can matter. So just as a reminder, we want to thank our friends at NSF for funding this work. Michael just published a couple of these papers just recently. He's pretty slow about writing, so if you see this video, Michael, you need to get those other papers done. And Ace, he's a good friend, my master's advisor, and then Dell and Lauren were undergrads that helped us. And actually, it's funny, so both of these are now vets, like veterinarians, so I have a lot of pre-vet students that come through the lab, which are awesome. Okay, so this is a question. So you're going to see a lot of papers. So those of you who are interested in ecology, I'm going to put a bunch of old papers here. Some of these are my favorites. They're not particularly long. Most cases are not particularly hard to read, but they're really fundamental papers. And so if you're interested in ecology, I encourage you to look at these papers, and you're going to read them and be like, this doesn't seem that interesting. But at the time when these were published, I really think they made a big impact. They still make an impact. Okay, so this was a classic paper, and it asked this question, why is the world green? And it basically comes to this, why do we see areas like this? There's so much out there that herbivores could eat. Why are herbivores not eating it? And I'm going to try to apply this question to lakes and ponds, because that's where I study. So just as a reminder, we think about these top-down effects. So again, I mentioned before, if you have fish, if they eat the zooplankton, then the zooplankton aren't there to eat the algae. And typically we think about the big zooplankton because these are visual predators. They're looking for these slow-moving Daphnia to eat, and if they're there, they eat them. The Daphnia have no chance to make it, and then the Daphnia aren't there to eat the algae. On the other side, we have this bottom-up approach, and the idea is that if you feed a system with nutrients, you get more algae. In this case, is a primary production. And again, these are nice data. This is a classic study. And you see that at some point this starts to saturate, and it's likely because something else is limiting. In this case, it's probably light. So algae need light. When they get so thick, it can be hard for algae to grow. So there's great examples of both top-down and bottom-up. So we're going to try to squish those together today. So this is the simple food chain. Again, the sun, phytoplankton, they get eaten by the zooplankton, which get eaten by the little fish, and then the big fish eat the little fish. So because of iCook and things, I tend to not work with fish. We do some work with fish now, but I try to stick with the invertebrates and the algae because it's much simpler. We just do what we want when we want to do it. And so that's what we're going to talk about today. So just keep in mind, I'm going to ignore fish for the most part. But fish are super important. And at the very end, you're going to get, if you like fish, you believe in fish being important, you're going to get you get to stand up later and cheer because I'll agree with you. But we're going to ignore them because it just complicates it. Okay, so you're going to see figures like this, and I want to make sure I go through this kind of quickly, quick enough that you can follow along later. So this question is, what's the effect of a consumer across a productivity gradient? And so this is going from low nutrients or low productivity to high productivity. And this is the consumer effect size. I like to do a meta-analysis. We think about effect sizes, so I'm sorry if you aren't comfortable with this, but it's just how I see the world many times. And the idea is that if you have no effect of the consumer, then as you increase production, there should be no effect. And we're going to call that resource controlled. But if the consumers have a bigger effect, so as you increase nutrients or productivity, you get a bigger effect of the consumers, and the line should go like this. This is what our predictions are. And these come from early papers. So Matthew Leibold's papers, they just spoke to me as I read them as a student. I encourage you, if you've not read these, they're awesome. They're so easy to understand. And so this is a figure of his paper looking at how you increase production, and as you increase production, you get more trophic levels because you can feed more organisms. And at the lowest level, you just have enough to feed the plants, but then you hit some threshold where you can now feed the consumers. But then as you increase production, you don't get more plants, you get more consumers. So this is the idea. You can see this alternating, like who's winning. So plants win, now the consumers win, now the plants win, and then so on. So in this particular talk, we're going to ignore the fish, so we're only looking at the phytoplankton and the zooplankton. And the idea is what happens in a system where you could have zooplankton, but you don't. And so we're going to look at this effect right here. So this is, and I'll show you what this looks like in a second. So there's some papers out there that talk about this. So this is one of Mike Pace's early papers between primary production and herbivory. And there's an think consumer effects do scale and do increase with activity. But what you see here on the y-axis is herbivory rate. And so there's some plants positive information to suggest with productivity, but it's not perfect. This paper done by Ace Arnelli, PhD work, and he did an analysis, and again, looking from low nutrients to high nutrients. You're going to see this increase to high nutrients. So if you don't like that, I'm sorry. And this is looking at the amount of algae without Daphnia, algae or above Daphnia, or yes, experiments. And you can find that in this case, this algal response factor, so this effect of Daphnias grows as you increase nutrients. It's a meta-analysis, not one experiment, but again, pretty definitive data synthesized showing that yes, as we increase nutrients, possibly the consumer effect gets bigger. Okay, so this is what Ace had, this is what he talked about as the algal response factor, just as a reminder. So you have a system where you increase nutrients. Let's say the nutrients are here, and if the prediction is if you had zooplankton, you would be here, but if you didn't, you would have the potential to be up here. So we're looking at that difference right there. And in this case, it's a ratio. And so again, this is what Ace showed. So again, pretty clear data showing there's a positive effect. So as you increase production, you get a bigger consumer effect. In contrast, people have looked at this in terrestrial systems and shown something a little different. And so this is John Chase's work showing as you increase rainfall, you get more production in terrestrial systems. And so then he's linking precipitation to this herbivore effect size, which is what we're actually kind of trying to get at. And you see this negative relationship. So in terrestrial systems, you see the exact opposite effect. Okay, so is it positive? Is it negative? What's the relationship? And so what's happening here? So these are the options now that we have. So initially, we go from low productivity to high. If consumers don't matter, you don't have any relationship. If you get a bigger effect, the consumers do matter. You'll find this positive relationship. But what John Chase talked about was what he called heterogeneous food webs. And what that really means is that you have defended plants. So these are plants that as you get more and more production, you have plants in there that aren't going to be eaten by the herbivores because they have some defense. And I'll show you some examples in a second. And so I'll just show you, maybe there's something unique about terrestrial and aquatic systems. So this is another meta-analysis, and again, talking about big herbivore effects or little herbivore effects. If you look at our two systems, we're talking about lakes or terrestrial systems. It does seem to be that aquatic systems have bigger effects of consumers. So there may be something that's special and different between these two systems. And then there's this really classic paper by Don. If you haven't read it, I encourage you to read it. I don't agree with much of what's in there, but it's a classic. You should read it. Now, not every paper you should read is something that you should agree with. It's just a good example of a classic paper. And in this particular paper, he has this table where he tries to talk about systems where you have these trophic cascades, where they typically happen and where they don't. And it's basically trying to link aquatic versus terrestrial. And he talks about all these things that, like species diversity, the habitat, the autotrophs, and I don't necessarily agree with anything in this table. So I don't think that aquatic systems have low diversity. If you ever kind of snooped around in a pond, you'll see tons and tons of phytoplankton, zooplankton, invertebrates, plants. It's incredibly diverse. So I don't really necessarily agree with much of what's in this table. But then he also argued that these plant defenses are rare in aquatic systems. And again, this is another example. It's not as well studied, but I think that you'd find these defenses there if you look for them. And just as a reminder, so my PhD, my advisor in the lab, we did a lot of work on plant defenses. So terrestrial systems, we've got these thorns, we have chemical defenses, the same in marine systems. But what about freshwater systems? So there's not as much done in the thinking about chemical ecology. But we're going to talk about cyanobacteria, also called blue-green algae. These structural defenses, so some of these can grow really big. Here's a picture I took with Daphnia that are around this culture of Microcystis. These colonies are humongous. There's no way that a Daphnia could put that in its mouth. It doesn't mean it could eat it over time by breaking it up, but it's not going to be able to take that and just pop it in its mouth and keep moving. And then these filaments can be really big. So I think that structural defenses are possible with cyanobacteria. Chemical defenses, there's hundreds of compounds produced by cyanobacteria. They're chemically rich. We know, literally something about a few handfuls of these things, but there's tons and tons of chemicals in these algae that we really don't have any clue what they're for. And some of these do have toxic effects, and I'm going to show you an example. So this is part of my PhD work where I synthesized the literature looking at the effect of cyanobacteria on population growth rate in zooplankton. And basically, it's really simple. We just look at the effect, the growth rate of the zooplankton on good food, this green algae or flagellates, versus bad food, which was a little bit or all cyanobacteria. And if this was an effect of 0, there'd be no difference, but there's a hugely negative effect showing you that cyanobacteria are clearly bad food for zooplankton. And people argued why that is, and I don't necessarily want to get into that, but we know that they're poor food. There are some fundamental... So we know... Okay, so now let's go tie this back into the ecology. So this is the figure that we saw a little earlier. This is a later paper by Matthew, 10 years later. This is the figure we saw earlier. Now what he's done with his second edition is he's added the theory of plant defense. So this is what earlier, but what happens with plant defense is as you increase production, at some point you have enough production to produce plants that have some defense. There should be some cost to having defenses. If there was no cost, then everyone would be defended, and they're not all defended. And so this is the theory, and then the idea is that once you have these inedible plants, they just take over. Okay, so let's talk about that with cyanobacteria. So there are some clear data showing that as you increase production, you get more of these bad algae. So the idea that these plants are poorly defended, or in this case are poor food but well defended, I think there's some proof to this. As you increase production, you get more and more cyanobacteria, which is, again, following what Matthew about consumer offense. This is not as well studied. In order to study the consumers, you usually have to have bigger systems, long experiments, but people are doing this work. And there's really classic examples showing you how consumers adapt to toxic or poorly quality food. And these are just some examples, but typically these are very specialized interactions. There's one predator, one prey. And but there's some really great examples. And again, this is just the one example that I think about, the garter snake and newt, and how the garter snake can eat the toxic newt, but then this toxic newt becomes a little more toxic, but then the garter snake becomes more adapted, and so on. So there's some really nice examples of this. So there's also a really nice example in the freshwater plankton world, and it's this Daphnia. Never work with Daphnia, you're really missing out. They're awesome. And so this is Nelson Harson's work from Lake Constance in Germany, and just briefly, they exploited the life history of Daphnia. This is Daphnia galeata. And typically Daphnia reproduce asexually, so these females produce females, and they produce more females, and it just keeps going. At some point, typically we think that the world gets stressful, they'll produce males which have sex with the females that produce these ephipia. And in the ephipium, there's 2 eggs, and then those will come off the carapace. They oftentimes will either float or sink. And what Nelson and his team did was they took sediment cores of different time periods of the history of Lake Constance, and then you can hatch these down. So these are resting eggs. You just pop them out of the sediment, you open them up, and then the Daphnia wake up, just like they did here yesterday. It's unbelievable. Took Daphnia from the 60s and 70s when the lake wasn't very productive, also when the lake had been really productive for a long time, and then more recently. And what they did is they fed them, again, good food, bad food. The zooplankton, phytoplankton people like really complicated, usually good food. And what we found is that before the lake got really productive, the Daphnia were not very resistant to cyanobacteria, toxic cyanobacteria. But then over time, there was this bottleneck. And so what was in the water column was more resistant. And so we think they adapted over time. And then as the lake got cleaner, some of these less adapted zooplankton kind of came back into the water column. And so, so this idea of this kind of local adaptation and rapid evolution over time in the lake is, honestly, this is one of my favorite papers. It's really short to read. I was actually sampling an aquaculture pond in West Alabama with a Mennonite farmer, Dan Clemmer. If you're watching this, I'm giving a shout out. And he knew more about this paper than my current grad students. And he's only read the abstract. But I was just talking to him about my work, and he was like, oh, yeah, he mentioned this paper, and I was blown away. This paper is so cool. If you like the idea of resurrection ecology, definitely take a look at this paper. But this is looking at these adaptations or evolution over time. So Ace Sarnelli and I, we did the same kind of experiment, but we looked across space. And so we took Daphnia from lakes that either had low nutrients or had low amounts of algae, and including cyanobacteria, or these really productive lakes that had really dense blooms of cyanobacteria. And we either gave them, again, good food or bad food. And what you find is when you take the average of the Daphnia from each lake, and then you take the average of those lakes, those 3 lakes of each type, you find that when you feed them this toxic cyanobacteria, the ones do better than the Daphnia from the clear lakes. And this makes sense, at least to me, that they're adapted. They've been in the system. They know how to deal with it. We don't know why they're special. The same species, but they are special. They're different. And again, I'll talk about these green lake Daphnias being tolerant to cyanobacteria and the clear lake Daphnia being sensitive. We don't know why they're different. We haven't figured that out, but there's something special about them. Okay, so back to the picture. This is where we do a lot of our field experiments. This is just one example. So we did the lab study to show that the sensitive tolerant differences exist. We wanted to go do the work outside to see if it actually matters. And so this is an example of one of our experiments, PVC frame. These are greenhouse plastic tubes that are sealed at the bottom. We fill them up with pond water. In this case, we actually used a pond that was next door to this pond because you can see all the mud, and a dense bloom of cyanobacteria. Again, 8-week-long experiment, 3,000-liter enclosures. We had experiments. The treatments are really simple. No Daphnia, sensitive Daphnia from clear lakes, or tolerant Daphnia from green lakes. And what we found is that if you add these Daphnia into these systems, well, if you don't add the Daphnia, they don't show up. But if you do add them, they get abundant. Both kinds of Daphnia live. They survive in these systems, but the tolerant Daphnia from the green lakes get more abundant, and it's reliably so. What happens is when they get abundant, the algae goes away. And they eat everything, honestly, everything. We have to end these experiments because we get algae growing on the sides of the enclosures, and then it starts affecting our numbers because it'll slough off. And what's really interesting about these data is that where we have sensitive Daphnia, we added them, they became somewhat abundant, an order of magnitude less than the tolerant Daphnia, but were abundant. There was no effect on the algae. So the Daphnia are in the system, they're living, but they have no effect. It's as if they're not even there, which is unbelievable. So the trait difference of these Daphnia dictates these huge effects on the amount of algae. Else the primary production, and so this is no Daphnia, sensitive Daphnia, tolerant Daphnia. And honestly, the production that we're seeing here is probably on the bag. There was okay, so we kind of do these experiments to kind of tease apart more interesting information. This is another experiment we did, similar pond, a bigger pond. We add a little bit less nutrients, but we had a system where we had either no Daphnia or Daphnia, and then we had added lots of nitrogen or no nitrogen. We do these experiments in the fall when it's not quite as hot, and when there's lots and lots of cyanobacteria. And in this case, what we find is, again, we have these huge effects of nutrients. So you add nitrogen, the algae explode, and then over time we find that there's a Daphnia effect. It takes a little while for the Daphnia to get abundant, and so this is showing you the amount of algae. So this effect we see here is nitrogen, so we didn't add nitrogen to these, but then it takes some time for the Daphnia to get abundant. What was interesting is that the cyanobacteria disappeared when the Daphnia were in there. although the algae was high. So in this case, there's lots of algae here, but it's not cyanobacteria. And then the microcystin goes away. Anyone who tries to do experiments with microcystin in the field, you find that it just disappears. It's just, anytime you want to study something, it goes away. So don't study. I guess it's just one of those things. It just happens. But the thing that I wanted to point out, which we were really excited about, was we took Daphnia animals at the end of the experiment and genotyped them to see who was there. And what we found, actually at the beginning, and this is what we loaded, we tried to have equal distributions, but we weren't perfect. And at the end of the experiment, we looked where we added no nitrogen, or just a little bit of nitrogen, and we found that it was basically the same distribution of clones that we had at the beginning, but where we added tons of nitrogen, where we had a huge effect of cyanobacteria, we found that one genotype just exploded, and it just dominated. And this one, we don't know why it's special, but it was certainly special. So the fact that you could have such kind of changes in basically evolution in these enclosures due to the response of a nitrogen addition was fascinating. Okay, so let's see. All right, so now tie this together, and I promise we're getting close. This is what we saw earlier. This showed you how consumers can adapt. They do adapt. So what happens? If you put this theory, what do you expect? I have no idea, honestly. I don't really know what to expect. That makes sense to me, but I don't know if it's right, but that's kind of what. So we think we would revert back to this if you have consumers that can deal with the plant defense. So those plant defenses don't exist. Okay, so I'm going to talk about two field experiments really briefly. And again, ultimately we want to see, does this consumer effect scale with productivity like Ace showed in his meta-analysis? And then honestly, the really cool part was if we do this across 2 seasons, do we find that, do the traits of the algae and the zooplankton matter? And this is the plankton. This is the experiment. Example, 5 ponds, a range of productivity, no Daphnia, sensitive Daphnia from clear lakes, tolerant Daphnia from green lakes. So this experiment was done in the spring. It's important because the spring is a time when there's not a lot of cyanobacteria. The water's too cold. The algae that's growing then is usually really high quality algae. And I'll talk about the fall experiment in a second. So this is, I like pictures and I like to make things simple. So this is what we'd expect. If there's no Daphnia, we get lots of algae. Where we have sensitive Daphnia, they should eat this algae because this is good algae. So they should eat all that. And then if we add our tolerant Daphnia, they should do the same. So we don't expect to see big differences, right? These Daphnia should eat the algae because the algae is high quality. And so this is what we're seeing. So this is the amount, this is the algal response factor I showed you earlier. So as you get bigger effects, there's bigger consumer effects. And tolerant, sensitive, or color-coded. And you can see that there's no difference. The Daphnia just love the algae. You give them systems with more algae, they eat all the algae. And this is just looking at Daphnia biomass. These slopes for the 2 different Daphnia are basically identical. So no differences. All right, so what about the fall? So this is where we really thought we would find something interesting, and we were lucky. So I talk about the super Daphnia. So at some point I want to get like Nat Geo, or probably not NSF, but someone to give me money to go travel the world looking for a Daphnia that could be heat tolerant and be able to eat like anything you put in front of it. So that's my goal. So if you know where these Daphnia exist, and you can send me some, I'd be happy to get them. But at some point I want to go search for super Daphnia. And the idea is I think these would be out there. I think there's some systems where we could find them. Okay, so this experiment, same kind of system, same kind of enclosures, same design, except we do it later in the year when cyanobacteria are abundant. And again, this is our predictions. The only thing that's really different now is for the sensitive Daphnia. These are Daphnia from clear lakes. They shouldn't eat this algae, and so it should be really abundant. And so this is what we saw earlier. Again, similar effects of tolerant, sensitive Daphnia. So what we found for the fall experiment was really different. And I put these slopes in here just to give you a sense of where the shape of the curve was, but they weren't significant. There's no significant slope here. There is here. So what we found is that the tolerant traits, those tolerant Daphnia, they have a similar but smaller effect on the algae in the fall than they did in the spring, but there's a significant pattern, there's a significant relationship. As you increase production, the tolerant Daphnia still have significant effects. So these adapted consumers can still deal with these prey that are not as good quality. The sensitive Daphnia, they don't have Okay, so now going back to here at the very end, why is the pond green? And ultimately, fish. And I've told the fish people in the house, you're doing good work, but fish matter. And but what's interesting is I think that at least in freshwater systems, probably in marine systems as well, that as you increase production, you tend to get a lot more small fish, planktivorous, and eat zooplankton. And so I think in our systems, you don't have lots of cyanobacteria because the zooplankton can't eat the algae. I think it's because the little fish are there and they eat the zooplankton that would eat the algae. So I think it goes back to the fish that we tend to ignore in our experiments. So I just briefly wanted to show you an experiment we're doing now. So this is the paradigm, at least several years ago when I was a PhD student, that zooplankton don't eat algae, this toxic algae. And then in West Alabama, where we're doing work, they add these shad. They think the shad eat the algae. They do not eat the algae. These eat zooplankton. They're very effective. And so what we're trying to do is do food web manipulations to try to promote zooplankton to clean up the algae. And it's working okay. I will say that it's not working as perfectly as we found. What we have noticed is that if you remove the little fish in these ponds, which are hyperproductive, is that there are native, or I don't know if native is the right word, there are zooplankton that live in those ponds that will become abundant and eat the algae. So I think this kind of system could work there, but we still have some more tweaking to do. Okay, so just as the conclusion here, the Daphnia, they can control these blooms of algae, but in fishless systems, so it may not be something that's like super important everywhere, but there are systems where this could matter. The thing that I think was really cool is that we did find that this herbivory effect was bigger than the effect of nutrients, which again, I'm a nutrient guy. I think that nutrients really matter, but the fact that in some cases consumers could actually squash the effect of nutrients. And then of course traits matter, and I think there's a lot of work lately showing traits matter. So this is just another example. Just as a reminder, I'm sure Allison has told everyone here, but we're organizing a meeting just around the corner. If you care about algae, you want to hang out with us, come see us. We'll be at the Perdido Beach Resort. And this is my contact info, so if you want to reach out to me, I'd be happy to hear from you. So thanks for being here. Thank you.